This is a typical problem of Mendelian genetics in which question is asked regarding the different types of gametes produced for a given genotype. So in this particular question we have to calculate, the very first step is to calculate the number of heterozygotes. Calculate number of heterozygotes. So in the given genotype, that is AA, capital B small b, capital C small c. So the number of heterozygotes is equal to 2. This is the one heterozygote, this is the second one. And we know very well that number of gametes for any given genotypes is equal to 2 to the power n. If I keep a value of the number of heterozygotes in the given formula, so 2 to the power 2, it makes 4. Therefore, given number of gametes for the genotype is equal to 4. Now this is another variant of the same problem. Here the question is asked regarding the recessive gene. We know very well that for any given genotype, the possible recessive gene is equivalent to all recessive genes of the given types of the gametes. For instance, in the given genotype, capital A, small b and small b, capital C, small c, capital D, small d, we are looking for a gametes with a genetic constitution A, B, C and D. Now this particular constitution is possible in the given genotype for only one gametes out of the total number of gametes. So the steps will be, the very first step will be to calculate the total number of gametes. Total number of gametes. So here again we will apply the same formula what we have learned in the previous slides, that is 2 to the power n. So here also the total number of heterozygotes will be 1, 2 and this is the third one. So while keeping the value 2 to the power 3, we get 8. Therefore, fraction of gametes with all recessive gene is equal to 1 by 8. Now there could be another variant of the same questions. Suppose if I replace the word recessive with a dominant one. If I replace the word recessive with a dominant one, then what will be our answer? So going by the same logic, for dominant, we are looking for the genotypic constitutions like capital A, capital B, capital C and capital D. So this genetic constitution is just not possible in this particular given genotype. The reason being that it has 2 small b. So the capital B is lacking. Therefore, fraction for dominant gene will be 0. Is equal to 0. Now again, this is a classical problem of Mendelian genetics in which a specific progeny for a given genotype is asked. So here we are looking for a genotype with a frequency capital A, capital B, small b, capital C, small c, capital D and small d. And the parental combinations for which we have to get it, that is a capital A, capital B, small small c, capital D, small d, and this is going to be crossed with a capital A, small b, capital C, small c, and capital D, small d. Now what should be our strategy? Since we have to get a genotype in which a very specific number of genes are required. For instance, we are looking for such a combination in which there should be capital A and capital A. Now these two capital A can be obtained by individually crossing these capital A and these capital A. So for gene A, following cross can be studied, that is capital A into capital A. Now this is going to produce this genotype. So required fractions. For capital A and capital A will be, so this is 1 out of the total 4, so this will become 1 by 4. Similarly, if you want to do for B, then again we have to study this particular cross. Now for B, we can cross like this, capital B, small b into, so this will give us the same genotype in equal ratio. So here required fraction for capital B will be, that is 1 out of 2. So this is 1 and the total number is 2. So that's why we have taken 1 by 2. So I'm sure now you can understand that for C, what we can take, we have to consider this cross. So if we consider this cross, that means we have to cross this small c into capital C, small c. So actually this is the same thing what we have seen in case of B. So this is also going to give you the ratio of 1 by 2. So the requisite ratio for capital C and small c, which is actually required over here, is again going to be 1 by 2. So far we got 3 ratios, 3 fractions in fact, 1 by 4, 1 by 2 and again 1 by 2. Now one last is left, and that is we have to get capital D, small d form again 2 fractions. So for gene D, we can write capital D, small d into capital D and capital D. So this is also going to give us the ratio of capital D small d and capital D and capital D. So again, requisite fraction for D is equal to 1 by 2. So if you multiply all those fractions, we will get the requisite fractions. So let's do it. So for A we got, so we got 1 by 4, 1 by 2, 1 by 2, and for the last one we also get 1 by 2. So requisite fraction will be that is 1 by 4, 1 by 2, 1 by 2, 1 by 2, that is equal to 1 by 32. So this fraction is our answer. Now here we are going to have a progeny with a typical genotype, that is capital A, small b and small b, and capital C, small c. Now the cross we have to study is a typical trihybrid cross. Okay, and this is going to be crossed with the same one. So again here for A we have to consider the cross between these A only. So again here if we cross, we will get so requisite fraction will be 1 by 4. For capital A, which is actually required, as you can see there, this is the requisite fraction. Similarly, if we talk about for B, we have to consider this particular cross, this one and this one. So for B, the cross will be like this. So again we will get this genotype. So for the given genotype, that is BB, this is this one. The fraction will be, again, this is the fraction we are looking for. So again it will be 1 by 4. Now for C, we have to consider this cross. So for C, again it will be crossed like this. So this is going to give this genotype. So requisite genotype as per the given question is equal to capital C small c. As we can see there, this is the requisite one. So that will be here So this is 2 by 4 is equal to 1 by 2. So finally, the requisite fraction for the given genotype will be multiple of the individual fractions what we got, that is equal to 1 by 4 into 1 by 4 into 1 by 2. Again, answer will be 1 by 32. So this is how such problems are solved. Now again, this is a very typical questions of Mendelian inheritance, in which the number of phenotypic class is asked with reference to any test cross. Okay, so first of all, let's consider this test cross. The moment we consider a test cross in which the hybrids or given genotype is going to be crossed with a recessive parent, we know very well. So this is the cross we are going to consider. So this is the cross we are going to consider. Now, this particular genotypic constitution is going to form 4 types of gametes. Again, here we will apply the same formula, that is 2 to the power n. So the number of heterozygotes are here 1, and this is the 2. So 2 to the power 2 is equal to 4. So altogether 4 gametes are going to be formed. As far as this genotype is concerned, this is going to form only one type of gamete, that is small a, small b, and small c. So let's see that what is the total number of genotypes that is going to be formed out of these combinations. So the 4 possible the gametes that is going to be formed by the parents are like this. This is ABC, then A small bc, then small a capital B C, and then small a small b and capital C. So all are going to cross with the same phenotype, so we can write like this: ABC for the number of genotypic class is equal to number of phenotypic class is equal to 4. What are the 4? This is the first genotypic and phenotypic class, this is the second one, this is the third one, and this is the fourth one. So what is the key learning? The key learning is that in case of test cross, the number of genotypic and phenotypic class are always the same. So that is why here we are going to get same genotypic as well as the phenotypic class. Or we can also write down that in test cross, number of genotypic class or phenotypic class is equal to 2 to the power n, where n is equal to number of heterozygotes. So this is the formula we can use for quick calculations. This is a typical Mendelian problem based on a dihybrid cross. Now a pea plant with a yellow round seeds is crossed with a plant with a green wrinkle seeds. And we know very well that yellow is a dominant over green. Similarly, round is dominant over wrinkle. So here small y is denoted with a green and small r is denoted with a wrinkle. So in the given case, what we are going to see that the progeny, that is yellow round, yellow wrinkle, green round and green wrinkle, are producing in equal number. So if it is producing in equal number, it's pretty clear that this is a typical case of test cross, because in the case of test cross only we get the phenotypic and genotypic ratio same. So it means the genotypes which is involved in making such cross will be a typical test cross. So the F1 hybrid will be like this, and the recessive parent will be like this. So if we cross, what we will get? This is going to produce 4 types of gametes, that is capital Y capital R, capital Y small r, small y capital R, and small y small r. This is going to produce only one type of gamete, that is small y small r. If we cross them, then we will get a genotype of YR. We can put like this. So what is the phenotype of this genotypic constitution? It is obvious, it is yellow round, similarly, what will the phenotype of this? This will be yellow wrinkle, for this one, this will be green round, and this is going to be green wrinkle. So it is obvious that all of this is going to produce in the ratio of 1:1:1:1:1. So it is a typically test cross ratio. So we know very well that in case of test cross, the cross is going to occur between the F1 and the recessive parent. So The very constitution of F1 is that it is heterozygotes, and in case of recessive parents, both genes are present in recessive forms. So this is the requisite answer. This is again a typical problem of Mendel, but for a change here, the requisite progeny with a given phenotype is asked. In the earlier case, we have seen that specific genotype were asked, but here phenotypic constitutions were asked for. So here we are looking for a tall, green and wrinkled plant for a genotypic constitution, that is capital T, capital T, capital Y, small y, small r, that is going to cross with a capital T, capital Y, small y, and capital R, small r. So this is the cross what we have to consider. Now let's concentrate over here that what will be the proportion of tall plant. Again, for tall plant, we have to consider a cross between these genes. Okay. So for tall, what we have to do? We have to cross TT into TT. So in all the cases, you will always get a TT that stands for tall. So their fraction is always equal to 1. Now for green one, we have to study a cross between capital Y, small y into capital Y, small y. So if we cross like this, we will get this genotype, capital Y. This is a typical monohybrid cross in which we get a phenotypic ratio of 3:1. So it is obvious that all these three will be green. So the proportion of the green for the given progeny will be, that is 3/4. Now in the next we have to discuss about, we have to find out the wrinkle one. Now this is the wrinkle one. What we have, this is the cross what we have to consider for the wrinkle one. So for wrinkle, mind it, here we are only considering the phenotypes. So this is the cross we will study, and after this we will get only two types of genotype, that is RR, R and small r, small r. So the total fraction will be 1/2, because the wrinkle in this particular case is only this one. Therefore, fraction of phenotype for tall, green and wrinkle is equal to 1 into 3/4 into 1/2, that is equal to 3/8. So this is the fraction, that is our answer. Now in the given question, albinism is the lack of pigmentation, we know very well, and that is going to occur due to recessive gene. Now a normal man and a normal woman is going to marry, but each of them is going to have one albino parent. So it means that man as well as woman both were heterozygotes. So the genotype of man will be like this. Similarly, the genotype of female will be again heterozygote, since they have a parents out of them, at least one of them were albino. So since this albino is a autosomal gene, so one of the parents is always going to transmit that gene to their children, and the children is normal. It means that this particular genotypic constitution in them is in the heterozygotes. So this is the cross we have to study. When we cross it, we will typically get this genotype. So out of this, these will be normal, but this child with this genotypic constitution will be albino. So the requisite probability will be is equal to 1 by 4. Now here there is a disease called phenylketonuria, that is an inborn error, and this is also carried by the autosomal gene. So a man and a woman each carries a mutant gene for phenylketonuria, but neither of them has this disease. Again, it means that both of them are heterozygotes. Suppose small p is a gene that causes phenylketonuria. So the genotypic constitutions of male will be again capital P small p, and for female it will be again capital P small p, since both of them is going to carry the mutant allele, but since none of them is going to suffering from them, so that's why they are heterozygotes. If you cross them, we will get genotype like this: capital P, capital P, capital P, small p, the small p, this. Out of these three, they will be absolutely normal, and this will be having a disease carrying child. So the fraction will be 1 by 4, or we can also write 0.25. that is the probability. Here I would like to emphasize that the question is asking about the second child. Important thing is that in case of a human, at a time only one child is born. So every time this particular event is absolutely independent of the previous event. So in nutshell, we can say that every event is independent. So even if the question is asking about first child, second child or third child, in all the cases the answers will remain the same. Thank you. For video lesson, you can subscribe to www.youtube.com/aptacats. You can also subscribe to www.facebook.com/toptrigger.com. We are interested to have your feedbacks. Please mail your feedbacks to info@aptacats.com.